Look at this cockroach leg. It's not even connected to its body, but it still moves when we touch it. Why does this happen? Before we start, it is important to understand what voltage is in the context of a cell membrane. Basically, membrane voltage, also known as membrane potential, is the difference in charge between the outside and inside of the cell, measured in millivolts. If the inside of the cell is more positively charged than the outside, the voltage is positive. If the inside is more negatively charged, the voltage is negative.
So now that we've introduced the concept of membrane voltage, we'll show how this voltage changes, which allows electricity to be passed through a neuron. This change in voltage is called an action potential. Action potential is very important because electricity moving through neurons allows us to move limbs and flex muscles. But in order to do an action potential, a cell must maintain a resting potential.
Let's look at resting potential. There are three things that affect resting potential. First is diffusion. Diffusion is a force that pulls particles from high concentration to low concentration. In the context of a neuron, there's a high concentration of potassium ions inside the membrane and a high concentration of sodium ions outside the membrane. Because of diffusion, potassium wants to move outside and sodium wants to move inside.
Now let's look at the second factor in maintaining resting potential, electrostatic forces. An electrostatic force is an attractive or repulsive force between ions. Particles with a certain charge are called ions. These ions generate a magnetic field of that ion's charge. So for example, a negatively charged ion will generate a magnetic field in a negative direction. When ions of the same charge are near each other, their magnetic fields will repel each other, while ions of opposite charges will attract. This means that ions of the same charge will be repelled by each other, but attracted to ions of the opposite charge.
The third factor is ion channels. There are channels all throughout the membrane that allow sodium and potassium ions to move in and out of the cell. First, there's the leaky potassium channel. This channel allows for potassium to always go in and out of the cell. However, potassium will rarely do this because, despite diffusion forces pushing it outside the cell, the positive charge outside the membrane will repel it back into the cell. In summary, the leaky potassium channel allows diffusion forces to pull potassium ions outside the cell, though few go there. It is important that there is a higher concentration of potassium ions inside the cell for action potential.
Then there is the gated potassium channel. This allows potassium to exit the cell during an action potential. Potassium has to do this so that the cell can be returned to its resting potential voltage of negative 70 millivolts. We'll show this as part of the action potential too.
Next is the gated sodium channel. This lets sodium enter the cell at the beginning of an action potential. When the voltage reaches its maximum, which is around 30 millivolts, a ball-like part of the channel plugs the gate so no more sodium can get in. When the voltage returns to negative 70 millivolts, the ball exits and the channel closes.
Lastly is the sodium-potassium pump. This channel allows for 3 sodium to exit the cell and 2 potassium to enter. In order for the pump to work, it needs ATP, the energy of the cell. The pump is used to return sodium ions outside the cell and potassium ions inside. Then it's back at resting potential and ready to do another action potential.
These channels, the diffusion force and the electrostatic force, all work together to create the resting potential. Remember, resting potential is when the voltage is at negative 70 millivolts and all the channels are closed.
Now for what happens next. In order for an action potential to happen, there must be a change in voltage within the cell. If a change no greater than 20 millivolts occurs, the threshold of negative 55 millivolts is not reached, and there will be no action potential triggered. A slight change will not have any effect, but a change greater than 20 millivolts, making the membrane voltage negative 55 millivolts, will trigger an action potential.
Once the negative 55 millivolt threshold is met, the action potential starts when sodium ions begin to flood into the cell through sodium-gated channels, which will cause the charge in the cell to peak, rising to almost positive 30 millivolts.
Now that the cell is imbalanced, it needs a way to restore itself. At this point, the sodium channels close and potassium channels will open. The Potassium channels will allow the high concentration of potassium ions to flood out of the cell, resulting in a voltage drop within the cell. This flood of the potassium ions out of the cell actually drops the voltage past its normal negative 70 millivolts, usually to around negative 77 millivolts.
To stabilize the cell, sodium ions will need to go back out and potassium ions back in. However, this means it will have to go against the electrostatic force. This will require energy. The potassium-sodium pump begins to move sodium ions out of the cell and potassium ions back into the cell. The pump operates by using ATP to move a protein, which will take 3 sodium ions and 2 potassium ions, and then moves them to their respective areas inside and outside the cell. The potassium channels also close again to prevent potassium ions from returning to the cell after they've been forced out. After a few milliseconds, then the cell will return to its normal voltage and the process will be ready to repeat.
When the action potential first occurs and the gates are triggered to open through the electrical signal, allowing the sodium and potassium to flood in and out of the cell, this changes the voltage outside the membrane of another cell, causing that one to open its gates, releasing its action potential. So on and so forth as the electrical signal travels down the axon.
Going back to the cockroach, the reason why its leg still moves even after it's been pulled away from the body is because it's connected to electrodes supplying power, and when the leg gets touched with a wooden toothpick, the leg sends a signal which triggers the action potential, which travels down the axon, activating muscles to move the leg.
Tetrodotoxin is a neurotoxin found in many marine species, such as the blue-ringed octopus and some species of pufferfish. The neurotoxin binds to the extracellular portion of the gated sodium channel. We'll explain why this is harmful to the nervous system and to action potential.
When sodium channels are blocked, sodium ions can't flood into the cell and raise the cell's membrane voltage. This prevents the cell from reaching its threshold voltage, meaning it cannot get its membrane voltage all the way up to 30 millivolts. This is a problem because it means electricity can't be transferred from neuron to neuron if the neurons are affected by tetrodotoxin. This causes paralysis because the muscle cells can no longer move. It can also easily cause death due to the fact that the afflicted person can no longer breathe either.